Good evening, everyone, and welcome to Genomics Lite. This is our Genomic Surveillance in Focus session with Valerie as our guest speaker. My name's Em. I'm the Education and Learning Officer for Wellcome Connecting Science on the Wellcome Genome campus, and I'll be sort of hosting and leading the session this evening. Before we kick off, if you've not come to our events before, we host all of our events with the aim of helping everyone explore what genomics means for our everyday lives. And we encourage you to join the session with a spirit of curiosity and participate in the chat box and ask questions as we go along. But we ask that you do so respectfully for the panel and especially for our speaker. Myself and Fran are here this evening to moderate the chat and moderate the session to make sure it's a safe and inclusive space for everybody. If you've not used Zoom webinar before, which is the platform we're using this evening, there should be a few buttons that you can use at the bottom of the screen to interact with us if and when you need to. So if you have any questions or comments or having any technical issues, leave those in the chat box and we'll try and help where we can. If you've got any specific questions for Valerie as we go through her talk this evening, you can pop them in the chat box, or you can use the Q&A box if you have that at the bottom. That just makes it a little bit easier for me to pull out the questions a little bit later. We've also got closed captioning available this evening. You can press the CC box and it should show a little black banner of captions. You can also move these around the screen. So if they're blocking anything on Valerie's PowerPoint, you can move the captions out the way. You can also get rid of them if you don't want them. So the three of us are all, well, we all work on the Wellcome Genome campus. We're all at home at the moment. And the Wellcome Genome campus is based just south of Cambridge. So we always like to know where people are joining us from. So if you can find the chat box and would like to, let us know where you are joining us from this evening. On the Wellcome Genome campus, we've got multiple scientific research organisations, with the biggest two being the Wellcome Sanger Institute and Embl EBI. As you can guess from the name Wellcome Genome, everything that we do on campus is somewhere related to genomes and genomics, with the Wellcome Sanger Institute doing a mixture of what we call wet lab experimental stuff, so working with DNA samples, working with pipettes, working in wet labs, and data science, whereas Embl EBI is solely concentrated on data science. I can see in the chat box we've got some people joining us from Scotland and from the States, so that's a really cool mix. Someone from London, so not too far away. Someone from Chandler's Ford, which I've not heard of, so I'm intrigued to know where that is, and someone from India as well. So welcome to everyone. Valerie's talk this evening will be looking at genomic surveillance. So it's useful if we start the session by thinking about what a genome is. So hopefully you might be sort of familiar with this picture up here. We've all got DNA, every living thing has DNA. And so a genome is the complete set of instructions within an individual or an organism that has all the information that organism needs to grow. So a genome is the complete DNA. So my genome is my complete DNA altogether. And Valerie will be sort of building on that idea in her talk this evening. So as a brief overview of what we'll be up to this evening, Valerie will be giving about half an hour talk on her on the topic of genomic surveillance. There'll be time for questions at the end, and if there's any questions that are super linked as we go through, we can also sort of, I'll chip in and ask them as we go. After that, we'll also hear briefly about Valerie's career. I know a lot of people joining us might be at that sort of A-level or upper high school age where you might be thinking about next steps, sort of university and beyond. So hopefully that'll be useful to give you an idea as to how Valerie has got into the career that she has. Again, there'll be a little bit of time for questions around there, and we'll be wrapping up about 5:45 this evening. This session is being recorded, so if you join, well, obviously if you join late you won't hear this bit. If you have to leave early, don't worry, you'll be able to catch up with that on YouTube and on our website as well. Lastly, before I hand over, Valerie has also written us some content. She's been very busy with us lately. She's written 4 awesome articles for our Your Genome website, all around COVID variants and genomic surveillance. So a lot of this she'll be touching on through her talk, but if you want to read more in depth on each of these topics, the links will be in the chat box in a second, and they'll also be on your Eventbrite follow-up email tomorrow, so you don't have to worry about grabbing them all this evening. that is everything from me. So I will stop sharing my screen. Valerie, I will pin you to the first screen and I'll let you take over.
Hello, everyone. There we go. Share my screen. As Em said, my name is Valerie. I'm a product lead on the COVID-19 genomic surveillance team. And for those of you who don't know, product lead is kind of like a scientific manager. So if you signed up from the website, you might have seen I was listed there as an advanced research assistant. And that is because a few months ago, the product lead position is actually a secondment. So it's a chance for me to do some stuff at a different level and kind of get a feel for it, as well as helping out with the genomic surveillance team, which needed people at that level pretty quickly. So to begin with, I'd like to do a poll question, kind of to get a bit of an understanding for how much do you know about genomic surveillance. So there's a few options here, which should pop up on your screen. And there's no right or wrong answer. It's just for me to get a bit of a feel for what do you know. Anything from nothing, never heard of it before this talk, all the way through to loads, because you're really interested in it and you've looked this stuff up before. If you just give a quick answer, and Em can let me know when we've got a series of answers from people.
So we've got a good mix. We've got an answer for each one. I'd say most people are sitting around the B to C range. I think most people have heard of it, maybe just because of the news. A few people are answering D, but yeah, mostly around that sort of B level.
Okay, that's pretty much what I was expecting, because it has been in the news lately a lot with the COVID pandemic. And there's been a lot of words that have been thrown about that only a few years ago, most of the public probably wouldn't have known about, something like variants and stuff like that. So we'll just go on. There we go. So what is genomic surveillance? So genomic surveillance, if you want a quick definition, is kind of involves sequencing the genetic material of pathogens and identifying changes linked to the origins or characteristics of a disease. Now, as I mentioned earlier, all pathogens have genomes. In most cases, this is DNA, but you could also have some, like COVID, which have RNA. But as long as it's genetic material, it can be sequenced. And the reason we do that is because genetic material tends to build up changes, mutations, as it gets reproduced. Now, if you're dealing with small single-celled organisms, this tends to happen when they reproduce or create more of themselves. With bigger organisms like plants and animals, you can have it A, for reproduction, but it can also be when you create new cells within the body. So at these times, you can have changes that happen, mutations. Some of these are inconsequential, some of them have pretty big effects, but these changes can be tracked. So that's why genomes are such a good tool to do surveillance on. And different organisms will have different rates of mutations. So when you do surveillance, you kind of know, is it an organism that changes very frequently with mutations or ones that don't? But genomic surveillance, part of why it's such a powerful tool, step which you see here in italics, the analysis. Because if you're just sequencing on its own, it doesn't mean a huge deal. You want to bring that information together with potentially information from hospitals and doctors or stuff councils can provide, kind of to get a feel for what's happening in that area. Are, you're suddenly seeing a spike in the number of hospitalizations, then you might be able to say this change that you saw in the genome is potentially causing that, because you can go back to hospitals and say to doctors, these patients have this mutation and those patients have the different mutation. And then you can start to see, do they have different outcomes? And that's where the power in the surveillance lies, is to kind of link all this information together, which can then provide information to doctors or governments to say, look, this is what's happening, this is why, and they can make decisions on what to do with that information. And part of the reason why we're able to do this now is we're in an era of genomes and genomics. And what that means is we're able to do it much more often than we used to. The sequencing technology is changing very rapidly, so we're able to sequence more genomes than ever before. Now, if you look at the bottom left-hand corner here, you see a photo of an old sequencing lab. It's not the best photo, it's a bit grainy, but that's because it's older. This is from the Human Genome Project. So back when we decided to sequence the first human genome, that started in 1990. and was declared finished in 2003. So it took over a decade to do. You see the machines were quite big, they're quite clunky looking. You see the old computer monitors, they're very heavy, very big. And that's where we were, 13 years to do the first human genome project. Now it's always harder to do the first sequence of an organism, but still we've come a very long way. If you look above that, the picture up top, this is the older sequencing area at Sanger. You've got the HiSeq X machines. They're still very big, but now they're sitting on tables. So they're smaller than they were. And instead of years, we're now talking days to sequence a human genome. So, it's a very big leap forward. I've heard people say that what was a PhD project, what they did their PhD on, has now become entirely automated. So it's kind of the technology's come that far. And the third photo on the right is the current sequencing lab at Sanger. It's, the machines are still pretty big, the NovaSeqs, but they can, now we're talking about minutes almost to sequence a human genome. So the technology's come a really long way. You can do a lot more sequencing of humans and all kinds of other organisms much faster than before. You can sequence them multiple times to make sure you've got a very good coverage, that there's no mistakes that have crept in. And this is why we're in an era of genomics, because you can now do a lot of sequencing. You can start to compare the genomes. And these are the machines we mainly have, but just to show you how far technology has come, this new photo shows the MinION, which is, as you can see, a handheld sequencing machine. Now, it does not have the power of a NovaSeq machine, but because of its size, you can take it out into the field. So you can sequence genomes live in the field, which has great advantages as well. So, MinION versus NovaSeq, which one you'd want to use kind of depends on what you're aiming to do, because you cannot drag a NovaSeq out into the field with you. They're very heavy. Some of these machines have required us to reinforce the floor. So, big clunky machine, but it does a lot of power. Smaller machine you take out to the field, kind of depends what you want to do. But as I said, this has really revolutionized our ability to do sequencing. Now, part of what we're looking for when we're doing sequencing with surveillance is to see, has the pathogen changed? And like I said, you can build up changes, but not everything means something. So what we're really looking for are variants, and that's when the pathogen has changed enough, the sequence of it, to affect how it spreads or how it behaves. So this is why we liaise with doctors and hospitals, because they can say, actually, we're suddenly seeing, the usual medicines aren't working anymore, or we're seeing a lot more people in the hospital than we were before. So if that ties in with changes in the genome, then you've got a new variant. Alternatively, you might see a lot more spread in an area, but the hospitals aren't being flooded, because not every change will cause a more severe illness. Some of it will mean the virus or bacteria, whatever your pathogen is, it'll spread faster and better, but it might not make people sicker. So this is what we're looking for when we're doing this type of surveillance. But one of the big types of variants we're looking for at this point is a variant that actually has changed enough so that it no longer, that the immunity given by a vaccine or a previous infection is no longer effective. So that we would call that an escape variant, because it's escaped from the protection provided. And that's one of the big fears we have with the current pandemic, is that this might happen. So when we're doing surveillance, we're always kind of keeping an eye out for this. So the way you might first suspect this is if you're starting to see a lot of people in the hospital who've been vaccinated or who previously had the virus, who are showing the same severity of illness as people who haven't been vaccinated. So that's one of the big things genomic surveillance looks out for, is kind of seeing, has this happened? Because if that happens, you may find that either one of the vaccines doesn't work anymore, in which case, countries might decide they want to start looking at different vaccines, or it might mean you're going to look at different measures. So you might want to do another lockdown or something like that. So that's what we're really looking for. Now, at this point, I'd like to ask an open question. So for this, use either the Q&A box or the chat box. But I'd kind of like to know, what do you think, when and how might we be able to use genomic surveillance, and what types of situations? There's not really a right or wrong answer here. And even scientists don't always know the answers to this, because sometimes we come across a new situation where tools like this can be useful in ways we hadn't expected them to be.
Someone has said to prevent another pandemic. So I guess as more preventative measure before something like the COVID pandemic happens again. Someone's put to track the development of new pathogens, public health and monitoring disease spread. Oh, a nice option, a nice idea of tracking pesticide resistance and tracking antibiotic resistance as well.
Yes, so if you know what you're looking for, yeah, you can start looking at the bacteria and see, have they changed in a way that might mean they're no longer, affected by pesticides. So yes, to track potential new things for a pandemic, you'd need to be monitoring the type of, virus or bacteria that could cause problems. So it's one of those ones, if you're tracking them, which some scientists do, then yes, you could get the early warning. Otherwise, you'd almost need to, find an area that's got some new disease and try and quickly get on top of it. But yes, those are all good options and they're all quite useful. So
we've also just had a question that linked to your slide about the different sequences. So someone said, with the improved sequencing, did the costs also get lower?
Yes, so the costs have gone down, especially if you think about it, if you're doing 13 years of work versus now you could only say weeks worth of work. Yes, it's definitely gone down. It's still, if you want very good high level sequence, it's still costs, but we're talking more around £1,000 rather than, I don't quite know how much the Human Genome Project costs, but it was definitely a lot of money because there were a lot of people involved, a lot of time involved. But yes, it's coming down, and that's partially why you can start to get the personal genomics. You can send out to get your genome sequenced by various companies, and that's because the costs have come down.
Awesome, thank you.
Now I'd like to do just 3 examples of kind of genomic surveillance being used in action, so that you can kind of get a feel for the breadth of it. Now, first up, we have SARS-CoV-2, which is actually the name of the virus behind COVID-19 or COVID. That's normally what you see it called in the media, but that's actually the disease. It stands for Coronavirus Disease 2019. And it's kind of, if you think of AIDS and HIV, one's the disease, one's the virus. So SARS-CoV-2 is what a lot of scientists use when talking about the virus, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. I think you can all understand why we want to go with SARS-CoV-2. As the 2 implies, there was a SARS-CoV-1, which was the SARS outbreak from a few years back. So that's to say it's coronavirus. The other one people may have heard of is MERS. And these are all coronaviruses. And on the right here, you can see a drawing of the coronavirus. And they're called coronaviruses because of the spike proteins. It kind of looks like the bits of a crown. So that's why it's coronavirus. So yeah, scientists quite often call things kind of as they look. It means it's very easy to identify them if you come across a new one. And now, like I said before, the sequence can quite often tell you something about the origins of a pathogen. So in this case, the sequence is quite similar to a coronavirus that has previously been seen in bats. So that's why a lot of people think it may have come in bats, but it probably came to humans through another animals. Some people say pangolin. We're not quite sure yet, but it kind of builds up mutations along the way. But because it's very similar, they're quite closely related to the ones seen in bats. And then once it hits humans, you start getting the human transmission. And that's where we're mostly focused on when we do genomic surveillance. But as I said, the sequence itself is very useful to kind of tell you what's come before it. But the basic sequence is also what's used with a lot of the vaccine efforts. So once we knew what the sequence was, those efforts could get going. And then we move on to the human transmission. So this lovely diagram was done by Alex Kagan, and it's kind of meant to show how interconnected surveillance can be. As you can see in the background, see the maps, you see splotches of different outbreaks, but there's also lines connecting them. And that's kind of shows you how the virus can transmit. As people travel, they can take one version of the virus to a new location, and there, it can change along the way or it can be similar. Then the green bit you see behind our archer is kind of showing, you can see it almost looks like a family tree. It's kind of to show how different variants might be related to each other. So some of them might be quite similar, some are much more different. So kind of how connected they are. But knowing which variant you have can let you fight COVID. And countries can kind of look around the world and say, okay, that country has the same variant we do, but they got it earlier. So how are they looking now? What did they do to try and suppress the outbreak, to prevent the outbreak? And if they were successful, the country looking elsewhere might say, let's do the same thing. If they look and go, oh, that didn't work, they say, okay, we're going to try something different rather than repeating the same thing. So it's kind of like, by doing this surveillance, you can tie together a lot of information based on what has worked, what hasn't worked, what could you try. If 5 countries have the same variant and they've tried 5 different things, and when a 6th country gets it, they can pick and choose what worked best and, kind of jump across all the things that don't work. So you can kind of make things a lot better. So another question, open question for you is, what do you think might be the weaknesses of genomic surveillance? Where and how can it fall down? I mean, it's quite a powerful tool, but any tool has its weaknesses. And if you use it incorrectly, it won't help. Now, this little graph you can see on the left shows the different waves of COVID-19 in the UK. And if you're not quite sure what the weakness might be, I'd say look at wave number 1 and compare it to the later waves and kind of think, wave 1, I mean, we're all from different countries, but the first wave, a lot of the world reacted very similar, had quite high levels in a lot of places. So, but the wave here on the graph is quite small. So does anyone have any thoughts?
Yeah, we've got a couple ideas coming in. So someone's put the gaps in sequences. So maybe there wasn't enough samples to know the full sequence of it. Someone's pointing out that for wave 1, there wasn't as much testing available. Having a look, mutations could appear at any time before we track them. So some might be more infectious with higher virulence. A few other answers for less testing in the first one. Someone's put the virus can mutate faster than we can sequence it, and someone else has put rapid mutation. So lots of good ideas.
Yeah, lots of good ideas. And the one about the lack of sequencing is quite important there, because yes, I mean, we're talking the wave, kind of the UK went into lockdown around March. While the UK has a lot of sequencing capacity, most of that capacity was already in use. At the Sanger Institute, we had various projects. There weren't sequencing machines standing idle. We had them for a reason. And building up genomic surveillance, especially on this scale, had never been done before. So you kind of had to get everything together to be able to start doing that. And the labs that had the sequencing capacity weren't necessarily the labs that had the actual samples from testing, which again wasn't being done a huge deal. So yes, it took a while to get everything together, but a lot of those answers show, what people were saying about not being tested is quite good, because it's genomic surveillance is great. It tells you a lot of stuff, but it only tells you, and this is the same across a lot of different areas of science, it only gives you an answer to the question you're asking. So if you're looking in one area, genomic surveillance will tell you about that area. So say you're looking at London. Well, you'll know what's happening in London. You might try and extrapolate it to Newcastle, say, but if you're not looking in Newcastle, you don't know what's happening in Newcastle. Now, you might be very, very lucky and it's the same variant. So what you've learned from London might apply to Newcastle. But unless you're looking there, you can't be certain you've got the same knowledge, the same variant. So what you're trying may or may not work. We've seen this a lot. Certain countries have a lot of sequencing capacity, so they're churning out a lot of samples. Some countries don't. So if you don't look at which variants are there, you don't know. Now, in the beginning, you had a lot of different names being bandied about for variants. At the moment, we tend to use the WHO label, which is something like alpha, beta, delta. Before that, you might see more the scientific names being used in the media, some like P1 or B117. Or the other one that was quite popular was location names. So the UK variant, the South Africa variant, Brazil variant, and in the UK variant was called the Kent variant. That's problematic because people will tend to say, oh, well, it originated there. But you can't actually know that. That's where it was discovered because those areas were looking. But it might have come from somewhere else that wasn't looking. Or it might be in a lot more places, but because a lot of those places aren't looking, you don't realize it's there. But once you start looking, you're going to potentially find more, but you're also going to know what you have. So as I said earlier, knowing what you have means you can then target treatments or stuff like lockdowns much, much better than if you don't know, because you can see what's effective against that particular variant. So yes, it's kind of to say genomic surveillance is a great tool, but you can't always extrapolate because different countries might have different variants and you don't know until you look. So the best way of putting together a really good kind of package of fighting it is to know what you have, because there's going to be certain things that work well against one variant, which may not work well against a different variant. And one of the ways we do that with the COVID surveillance team I work is we actually use an algorithm that tries to make us pick samples from across the UK. Now, we don't always know where each one comes from. The machine knows that, the algorithm, knows that, so it tells us. And if one day we don't pick some from a particular location because the samples weren't great, it'll make sure the next day we sequence some from that area so that we have a full coverage, which lets us know as soon as possible if something unusual is popping up. Now, if you're getting, higher numbers in one area, you get stuff like surge testing that will then get fed back to us, and we might get priority samples from, say, someone who's come back from a country that's got a high virus rate and has come up positive. So those will get priority, but we still want to do as much sequencing from across the United Kingdom as we can, so that if anything new pops up, you kind of know as soon as possible, and that you can tie it in and go, actually, this is a different variant. We need to know how it's behaving. Is it, more virulent? Is it more lethal? Sometimes it'll be more transmissible, so more people will get sick, but it won't actually cause a worse disease. So you can't assume that a mutation that's taken off is going to make it more deadly. It might just be better at passing between humans. So, but again, you don't know until you look. Now, a second example I want to show is talking about Ebola. Now, in this case, the virus and the disease are both called Ebola. It's a hemorrhagic fever, and depending on which variants you have, the lethality of Ebola can vary quite significantly from, say, 60% to over 90%. So which one you have can make a very big difference to how the virus has an effect on people. Now, Ebola springs up sporadically across Africa with little warning. This map kind of shows various areas that have had outbreaks, the years it has had. Now, the big one we really heard a lot about was the 2013 to 26 outbreak, but there have been outbreaks since that just haven't made the news as much, and there were plenty of outbreaks before that. Now, genomic surveillance can be used to help determine patterns of transmission in these cases. So when you had the bigger outbreak kind of across Sierra Leone, Guinea, and Liberia, when you had new cases, if you sequenced, you could see, was it part of this existing epidemic that was happening in that area? Because since Ebola springs up quite often, a second version could spring up in the same epidemic. So if you get a case where a variant is very different, then you actually people can realize they've got a second problem in the same area, and you'd have to track back where that person got it. Whereas if they're in that area and they get the same version, you still try and figure out who they caught it from, but it's, it's part of an existing epidemic. The other way it can be quite helpful if someone in one of the neighboring countries suddenly tests positive for Ebola, you want to sequence them to see, is this a new outbreak? Is this a different Ebola? Or is this the same, part of the same epidemic? Because if the person says they haven't been to the countries where the epidemic happens, either is possible. So if they test their virus comes back as being part of the epidemic, then you'll try and see, okay, if they've not been to the affected area, how did they get it? So you kind of look do they know who might have been there? Because you want to figure out the route of transmission. Because if you've got one that's 90%, has a 90% lethality, then you want to catch everyone who has it in between. Now, like I said, genomic surveillance in terms of medicine is a relatively new tool, and it's still showing us surprises from time to time. So the 2013 to 2016 outbreak had a lot of surveillance, and that became useful unexpectedly earlier this year because there was a new outbreak in Guinea. And when they sequenced it, they were quite surprised to find it was almost identical, the sequence, to the 2013 to 2016 outbreak, which means it's not a new, doesn't come from a new origin. And it was so similar that it having lain dormant or passed through various animals and then come back to humans was less likely. So they actually went looking to the survivors of the first outbreak because it was so similar that it looked like the same virus. And they actually found that one of the people who had survived it had become reinfectious. So, something that in the area people had always worried about, and survivors would sometimes face quite a bit of stigma. And now they had a documented case. So this means it's opened up new avenues of research so that they can look at, how does this happen? How does Ebola lay dormant for so long and then suddenly become reinfectious? Which is quite important to know because, there are quite a few survivors out there, even with that high of a lethality rate. So that's led to new avenues of research and meant that they could focus much more quickly with that new outbreak on what might be happening rather than thinking it would, it came up from just sporadically popped up again. Now, my third example is for MRSA, which stands for methicillin-resistant Staphylococcus aureus. So hence MRSA. This is a bacteria, and it's responsible for a lot of very difficult to treat infections because, as the name suggests, it's gained resistance to drugs. So it's quite common in hospitals, nursing homes, prisons, anywhere where you might see quite a few wounds and open wounds. And so hospitals are quite vigilant for this because if it appears, you want to get rid of it as soon as possible because you've got a lot of people who either have reduced immunity or if like babies who have no immunity yet or very little immunity. And the reason I bring this one up is because this is actually quite a small scale outbreak. And it's to show that this type of, outbreak can use genomic surveillance too. So in this particular case, the outbreak happened in a maternity ward. And it showed quite an unusual pattern of transmission. So normally when you have an outbreak like this, you can't always figure out if it's coming from the community or how it started. But once it starts, it tends to pass between patients. And you kind of pass from one to the next until you break the cycle. And this is quite often done with deep cleaning, decolonizing people who have it. But in this case, they had a bit of an outbreak and then they might have had like 17 days and then there was another outbreak and then you might have another month and then another outbreak. So that's quite unusual. And their regular procedures hadn't quite worked and looking at, where it was coming from wasn't working. So actually sequencing was brought in for the first time in this type of setting. And they kind of looked at the samples that had been kept and they're saying, yes, actually, this is the same outbreak. And because the usual methods hadn't worked, they kind of got permission from all the staff to check them all for MRSA, and 3 people came up as having MRSA. And when those were sequenced, one of them was found to have the same version as the outbreak. So this doesn't tell us whether that first staff member brought it into the hospital or whether they caught it from one of the first people to or one of the first babies to have the MRSA. But regardless of how it started, that person, not realizing they had MRSA, was unwittingly passing it on. That's why you had those breaks. You know, they cleaned everything, they stopped the transmission, but then because the staff member had it, they were giving it back every now and then. So it was the first time you had sequencing used like this in the hospital setting, but it was able to kind of come in, confirm the connection. They had actually identified a few new cases and to help uncover the source. So that kind of, I wanted to include this one just to show, now it doesn't have to be a global pandemic for genomic surveillance to be quite useful. So just kind of a bit of a recap, say, genomic surveillance is quite a wonderful tool to track the changes to these pathogens and their effects, but it's got the limitations we discussed. And as a result, as with any tool, it needs to be used appropriately. The answers you get will be dependent on how you use the tool. So kind of like make sure you ask the right question if you want a specific answer. But also we're still in the learning stages of what genomic surveillance is. So what we can do with it and how we can use it is still being, you know, we're still discovering it as we go. We've learned a lot of lessons from this, you know, using it globally like this. And, you know, we're probably going to improve as we go. But it is quite a good tool that has helped us a lot. So does anyone have any questions on this bit so far?
Thank you so much for that talk. I've learned a few things through that. There was definitely like the Ebola sort of become really infectious. I had no idea, so that was really interesting. Someone's asked sort of more generally about transmission. Is it possible for pathogens and viruses to travel through the air? I think this was asked during the Ebola section, but I guess it sort of applies to all of them.
The answer is both yes and no. So it depends on which pathogen we're talking about. Something like Ebola is more transmitted through blood and, you know, fluid, bodily fluids type stuff, but some of them are more transmissible through the air. So something like COVID tends to be, it's more in aerosols and droplets, but if you're standing close enough together, those can transmit from one person to the next. But yes, there's a lot of, you know, with Ebola, one of the big fears is if it ever will become transmissible through the air, it would be a very, very big problem because of its very high lethality rate. So different pathogens have different means of transmission. So it kind of depends on which one you're talking about. But that is part of why a lot of the pandemic preparations had focused more on flu-like pandemics, because that was a type of transmission that we were quite familiar with. And the last big pandemic had been a flu pandemic. So it's kind of where people were alert for, but it would depend a little bit on which pathogen you're talking about.
Awesome. We've got another question saying, do you also genetically sequence the individuals that build up natural immunity? Does that help with pathogen protection and seeing how viruses will evolve in the future?
This is one of those areas where now that we can sequence human genomes a lot faster, it would be quite interesting to do. Now, our team at Sanger isn't involved with this. We're very much focused on the COVID sequencing, but if you start to find people or groups of people who are immune, it would be one of the things would be to look at the genome. It might not be the only thing, because maybe their protection is coming from something they're doing environmentally or behaviorally. But if you look at the genomes and you're finding actually this group of people does have differences with people who aren't immune, that could be very interesting, because that could lead to new ways of creating protection or vaccines. And even if you can't replicate it, it can quite often provide a lot of information on how that protects them, which then might give us ideas for different things to try. So yes, definitely with the increased capacity to do sequencing, that's something that could become much more prevalent if we have growing groups of people who are immune.
I think I definitely saw some sort of adverts sort of last summerish that were asking for the people who had lived with somebody who had COVID and despite living with them hadn't got it. So I think there's definitely that sort of testing happening. It's not as Valerie said, it's not something that we are doing on campus, but I'll see if I can find any links to those. So I don't know if any results have been published, but if they have, I'll try and find some and pop those in the chat box in a second. Someone asked a great question saying, would data sharing between countries be more important than before to successfully track the evolution of things like COVID?
Absolutely, because if countries aren't sharing, then they'll know which variants they have, but nobody else will. And that means that nobody else could learn the lessons from that particular, what they've tried, that country, what they've tried. So data sharing is very, very important. And actually from the first sequence that was done of the COVID genome was published by the Chinese scientists that did it. They published it, which meant a lot of companies working with potential vaccines were able to get going very, very quickly. That's part of why the vaccine efforts have come along as quickly as they have. But yes, with this type of thing, the more data that you can share, the better, because some countries will have much more ability to churn out sequences than others, because some countries will only have tiny sequencing capacity. And like I said, you won't know everything that's going on in their countries, but if everything they have found indicates they've got the same variant as another country, they can already start to learn lessons. As I think a lot of countries are trying to roll out more ability to sequence, but it does cost a lot of money. Some of those NovaSeqs, they go for a lot of money, and you have to set up the entire infrastructure. But yes, the more we can share what we're finding, the faster everyone can adapt. So sharing the information in cases like this is very, very important.
I guess as a good follow-up question, someone's asked, is there a global consensus on COVID surveillance at the moment?
I have to admit, I don't know as much. I know what we produce does get made available, and there are lots of places where they do pull information together, but I don't know if like every last country is sharing at the same level. But a lot of what I've seen does seem to indicate a lot of countries are sharing, because you do quite often see graphs and talks about, this country has found this country's found that. So I think in this case, a lot of people are sharing just because it is so valuable and it is a global pandemic. It's not like certain areas have managed to really keep it all out. So it's affecting everyone. So if the faster we learn about new variants, the faster you can act on them. Because as we've already seen, like the Delta variant, it went global very, very quickly, and it's affecting everyone.
Yeah, I think a lot of countries definitely are sharing data, whether it's more sort of voluntary than as a global consensus, but that might change in the future once we see how important that is. Just having a look for other questions. We've got a couple that are slightly less on genomics surveillance specifically, so I'll still ask them, but obviously we're sort of moving slightly off topic, which isn't a problem. I don't know if you know the answer to this, but someone's asked whether there's any new information about vaccine efforts for Ebola.
I had heard there was something they wish to look into more. The biggest problem you have with Ebola, unfortunately, is because it springs up all over the place, it's very difficult to put a study into place, because the best vaccine studies, you kind of need a large, people vaccinated and people unvaccinated, so you can kind of see what happens to people who are vaccinated. But if you don't know where the next epidemic or the next, outbreak will happen, it's very hard to get that group of vaccinated people to kind of compare to the unvaccinated people. So that they're doing their best efforts, but it's one that's quite tricky. So I know they were working on it. I don't know more about if they've gotten more successful, but yes, because of the sporadic nature of where it pops up, it's harder to do than, say, with COVID, because everywhere had COVID. So you could kind of vaccinate people in one area, and to know if a vaccine's effective, you need a large number of people who've been vaccinated and unvaccinated to kind of compare the results, kind of saying, okay, actually, so many % of the vaccinated people have or haven't had it. And what's that compared to the unvaccinated people? So that's the problem, is getting that group that's vaccinated in an area if you don't know what area is going to have Ebola. Because obviously if we knew that, we'd kind of try and stop it in that area.
A really good question that yeah, I'm really interested to know your thought on is how much worse do you think the pandemic would have been without this sequencing effort?
That's quite hard to say. I mean, if you talk about any sequencing, then you've got to say without that initial sequence, the vaccine efforts would have taken a lot longer to get going, and or the study behind the research that led to the vaccines. So that could have caused quite a big effect if we had those delayed. But it's, I mean, it's a bit hard because I don't know what every country's doing, but like with the UK, sometimes the results of going, actually, this new variant is spreading quite quickly, means that they've locked down differently than they might have. So if that's been happening globally, that could have changed, you know, how many people died. Because if you're realizing something that's more transmissible and more lethal is springing up in your area, I think a lot of governments would be more likely to go, we've got to react quite, you know, in a big way. So it's hard to say, but my guess would be it probably had quite a big effect if the results have been acted upon. Because that's one thing, you can do all the surveillance you want, you can provide a lot of information, but if you're coming up against any group that decides they're not gonna, you know, that it doesn't matter, they're gonna do what they want anyway. That's another actually potential weakness, is if it's not acted upon or if it's acted upon incorrectly. So I guess if somebody with an agenda takes the data to try and twist it around, that could mean it doesn't work in the best way possible. We have that with any kind of information. You can always twist information around to suit different goals.
We've got a question that's linked to your answer about Ebola earlier, saying, can we predict which pathogens have a higher chance of causing an epidemic or a pandemic? Like how you said, Ebola, if it became airborne, would be that much worse.
Well, it's easy to imagine scenarios where something becomes a lot worse. Whether that's evolutionarily likely is a bit of a different story, because it kind of depends on the circumstances. Now, I think one of the reasons why a lot of people were worried about a flu pandemic is because of things like battery farming, because we quite often with battery farming have a lot of, say, chickens in a very, very small place. So if you have huge numbers of animals in a very small place, and if ill animals aren't quickly pulled out, so they're kind of all kept together, you've almost condensing evolution kind of going, here, spread very quickly over a large area of animals. Or if you've got people moving into an area that's, you've got exposure to wild animals that we haven't had before, you open up the opportunity for stuff from, say, bats to cross to humans that maybe normally, you know, we normally wouldn't have been in the same space, so it wouldn't have crossed to us. So there's definitely ways that, you know, certain places where it's more likely to happen, but that doesn't necessarily mean something won't come out of nowhere unexpectedly, because there's still so much we don't know about the natural world. And, you know, we're still discovering all kinds of new pathogens as we go. So there's certain ones that are more likely, but the ones we don't know about could be even more likely. And if they encounter stress we didn't predict, they might change in ways we didn't predict.
I guess it sort of links to one of your first slides that this coronavirus, we've seen other coronaviruses before. So again, there's like pockets of viruses that maybe we're aware of more. And so, as you were saying, the more you look at that one thing, the more likely you are to spot it in that one thing. Whereas if we're not looking at a different pathogen, we probably won't see it coming.
Exactly. I know there's some people are more worried about than others, but again, that doesn't necessarily mean it will happen. It means they've got a higher likelihood. We're talking about coronaviruses. Some of the pathogens that cause the common cold are coronaviruses. So, you know, we've got some that are already among us that don't cause, you know, most people just get the common cold. So it's sometimes hard to predict. Whenever you get to any living thing or anything that can change with that type, it can get messy.
We've had a question about one of the last graphs you showed on your slides, asking on the graph you showed of SARS cases, do you think we'll reach a point where we can no longer create vaccines quick enough to protect against new variants?
It's one of those things where it's possible, but SARS-CoV-2 has been actually, given it's an RNA-based virus, it's actually surprisingly stable. So that's actually a very good thing. I mean, we're seeing these variants, we're seeing these changes, but actually RNA viruses normally change faster than this one does. So the fact that it's so stable is actually quite good for us. And just because it's changing doesn't actually mean it'll always get worse. It can change to become better from our perspective in that it can change so it doesn't make people as sick. Because if it's not killing its host, that's got advantages for a virus as well. So just because it's going to change doesn't mean it's necessarily going to be bad for us. I mean, if we were really lucky, it might change to become much less virulent and much less lethal. I mean, it's unlikely to happen, but it's a possibility. So, and for it to suddenly speed up and change a lot constantly, I mean, it's possible, but I'd say it's probably less likely.
And I guess also in the case of this pandemic, we've got a few different vaccines as well. So as you were saying earlier, if it mutated in a way that one of them was less effective, hopefully other ones that we have available would still be effective. So I guess we're somewhat lucky in that. It always feels weird saying lucky in regards to a pandemic, but in this case, specifically having a few different vaccine options, I guess is better than only having one.
Yeah, absolutely. And some of these vaccines are new types of vaccines that are more easy to change. It's like if you get the flu vaccine every year, that tends to be a different one every year. And the way some of our COVID vaccines have been made means they can be tweaked much more easily than previous vaccines could be. So that's a help. Whether they could be tweaked every time, we won't know until we have to try it. But the fact they can be tweaked more easily, puts us in a better place to start with.
Awesome. Those are all the sort of like key questions really linked to your talk. So since we're sort of at 5:20, do you want to give us your sort of brief history of your career? If people have any more questions about the talk, keep putting them in the chat box and Q&A. Me and Fran will reply if we don't have time to get through them all. But you can also add any questions about Valerie's career as she's talking through that as well.
Okay. So quick facts for me. So I was born in Belgium and we moved around a lot when I was young. So I've lived and been educated in Belgium, the USA and France, and it's more than once in every country. When I got to the end of high school, we were living in the States. So for uni, I was kind of looking there. And I went in the end to the University of Massachusetts at Amherst. There's the logo you can see here. And I did 2 degrees there. One was Bachelor of Science in Biology. And when I did that one, I did do quite a heavy focus on genetics because this was around the time, my first year uni, when you had the Human Genome Project results coming in, people announcing it's complete. So there was quite a big, a lot of media talk about genetics. It was quite interesting. We just, starting to talk about what the fact that we had a human genome could do. So I did as much biology, genetics as I could. Some of it was, the basic genetics, the lab work. Some of it was into cancer genetics and stuff like that. But I also had quite a big interest in neuroscience. So my second degree was Bachelor of Science in Psychology, which was on the neuroscience track. So I did a lot of neuroscience, neuroimmunology. And in order to graduate with honors from the university, you had to do a thesis project. So I worked in a lab that worked with MDMA, which is the active component of ecstasy. And we were looking at rats. So how did it affect the brains of rats? And my project was actually mimicking teenage use. So kind of a few days of nothing and then 2 big doses, kind of like a weekend use. And then looking at what does it do to the brains of these rats. And then after that, when it came time to find work, I did actually struggle a bit in the beginning because it was, late 2016, 2017, so not too far off the big economic problems. And I found one at the Wellcome Sanger Institute. So I joined the mouse genetics project. So it was kind of set up to learn more about genes because we're actually really bad at predicting what genes will do. So we were working with mice that had one gene knocked out. So the gene was made to not work anymore. And we would test these mice on all kinds of tests next to normal mice and kind of see how do these mice differ. And we were looking at, metabolic areas, skeletal areas, the senses. So we were really trying to figure out as much as we could about what each of these genes do. It was part of a global effort to do this for every single gene in the mouse. And during the course of this, because I did this for quite a few years, we also were looking at areas, like I said, you kind of looked at the brain, kind of looked at developmentally and bone. So it was quite a big project looking at quite a lot of different things, which I was very interested in, because you might be running the same test, over the course of several years, but depending on which mutations you had, you might see some very different effects. And then this ended when the Sanger Institute decided to close the animal house. So I ended up looking for a job in the middle of a pandemic, because it was, summer last year. And I moved across at Sanger, actually, from mouse pipelines to DNA pipelines. So into the sequencing world more. And my initial project that I was involved with was the UK Biobank, which actually kind of connects back to some of the questions we had earlier. It's kind of this type of project where you're looking at a group of volunteers and kind of following them over a number of years. So the project can kind of look at health records. They do surveys to see how are the participants doing with diet and exercise, kind of what lifestyle are they living. And the bit we were contributing was actually sequencing their genomes so that they can then take all of this information together and kind of say, okay, at the end of so many years, some people will have developed various diseases. And it's kind of like, can you piece together the information about, the environment, their genetics, and kind of see, is anything popping up as potential flags for maybe being connected to those health outcomes? So to kind of try and learn more. So it's not a big data area, and these types of projects require a lot of bioinformatics at the end of it as well, to kind of draw all the attention together. And one of the projects I was also a little bit involved with at that time was what we called internally Project Heron, which was what turns into the COVID-19 Genomic Surveillance Program. And as I mentioned at the very beginning, at the moment I'm doing a secondment. So when they were expanding, because they started with 10,000 samples a week, 20,000, we're currently at 40,000. And the goal is to increase quite sharply how many genomes we sequence a week. So they needed kind of more the management level, and a lot of people who are already at that level, if they're coming from other companies, they have to give longer notice periods before they can change jobs. So they were looking internally if anyone wanted to do this as a secondment. So that's kind of how I then moved across to Project Heron to help out there. And that's where I am at the moment. So yeah, I guess any questions with that or anything?
One question that I always like to ask our speakers, knowing that we have a lot of sort of upper high school students in the audience, is what sort of made you pick, of all the things you could have picked at the end of high school, why biology, why neuroscience? Was there anything particular that really made you want to do that specifically?
Well, because I did, the high school in the States and uni in the States, it's a bit different from in the UK. You kind of don't really have to, with the A levels and GCSEs here, you have to start picking what you're doing. Whereas in the US, kind of to get into the better universities, you almost have to do a bit of everything. So I was still taking a lot of courses towards the end of high school across a wide range from history to biology. And one of the courses I took the last year was psychology. I kind of taken it, I have to admit, because everyone was taking it. But that's where I got introduced to neuroscience. And I have to admit, that's what really pulled me in and kind of made me very much focus more towards biology and psychology, because before that I had a lot of interest in English and history. But because the science there of kind of like how the different bits in the brain could affect so much of, how you're thinking, but also just the biology underlying all of it, really intrigued me. So when I was looking at the universities, I kind of very much wanted one that had this type of program to continue. And then the genetics component came in a little bit later. Like I said, there was a lot in the media about it, because when I went to university, genomic surveillance didn't really exist the way we do now, because the sequencing capacity has increased so much over the years. And you still see it. There's different things cropping up with genetics all the time that we didn't really think about. We've got now a very big ethical area that's opening up with genetics, kind of going, just because we can, does that mean we should? And I think that ethical area in genetics will actually expand quite a bit as we deal with new legal and ethical questions that crop up with the fact that we can do a lot more with genetic data.
Osman, actually, as you said that, it reminded me that the next session we'll be doing on this is around CRISPR, which is a genome editing technique. So that definitely, of all topics in genomics, is one of those areas that definitely is a just because we can, should we? And you might have heard stuff in the news about CRISPR babies a few years ago. So we'll be touching on that in our sort of next session around that. As you mentioned, the UK Biobank project that you worked on, someone's asked, how stringent are data protection policies around human genome data? Is the individual data made anonymous, and is this beneficial to the donor? How is their data shared? So I don't know how much you know about this, but from your work on Biobank, I imagine you will have touched on it a little.
Well, what I can say is we never knew who people were. By the time we got samples, it was a number. So somewhere that number in some database relates back to a person, but it's not information we have. But in general, the levels of scrutiny or how well it's protected will depend country to country. There's probably some countries that are much, much better at keeping that type of stuff anonymous than other countries. Normally, if you're using this type of information in research, it tends to be quite anonymous, but like I said, it will depend a bit on different countries. As to whether it gets back, that's actually sometimes quite contentious. A lot of the science that's done with this, it wouldn't be reported back, A, because you don't have the good routes of taking the information back, but also because it's, you know, say you find somebody has an increased likelihood of a disease or metabolic disorder, there's a lot of discussion going around about, well, should it be fed back? If so, what level of percentage? You know, if it's a 2% increased risk, is that worth reporting back? If it's a 90% increased risk, is it more? You know, there's still a lot of discussions going on about that. And there's also a lot of differences as to whether people want to know. You know, you've got certain disorders, genetic ones, that it's kind of like if one of your parents has it, it's a 50-50% chance whether you will have it. And there's quite a lot of people who decide they never want to get tested. They don't want to know. So a lot of the answers regarding information being fed back are very subjective. And quite often the scientists aren't genetic counselors. So it's also sometimes like, what's the best way of feeding that back? Because you need an infrastructure in place to make sure it's done properly so that the person getting it back understands what it means. Because if you're talking about small percentages, does that have any real impact? And if it's something that's actually quite bad news, they need to be supported and have the proper support network if they're receiving that information. So it's an area that's, I think, still very much, they're doing a lot of research around how to do it, whether we should do it, whether we shouldn't do it. But I think there's going to be a lot of work around that in the coming years.
As you were saying about sort of different countries obviously having different rules around this and different laws, reminded me of a study that one of our colleague teams have been running called Your DNA, Your Say, where they've been looking at people's views in different countries. I'll pop that in the chat box for anyone that's interested, but essentially not only do different countries have different laws about it, but citizens in different countries have different feelings about who should have access to what data. So again, it's definitely one of those, how should we manage data, like, you know, what do the public think, all of that sort of stuff that's really important to think about the more we do things like human genome testing and genomic surveillance of anything relating to humans. Had a couple of sort of linked questions around that sort of hereditary and genome testing of people. Someone said, I seem to remember a proposal around having all babies having their genome sequenced at birth and then that data being on their health record. Do you personally see that as a direction that health genomics is taking?
I think I'm not 100% sure, but I think in England or the UK, what they tend to say is you can get prenatal testing if there's fear for certain conditions, but the moment the baby's born, unless it's a condition that affects childhood and has a very severe risk of childhood, you cannot do it. You have to wait till the baby's 18 because they need to have their say. It's their information. And, does anyone else have a right to know that they might have an increased risk of cancer in their 40s? It's kind of like it's their information. So it's that kind of, and different countries probably won't do this. Some countries might say, yes, let's do it, let's share it, but there's a lot of information there. And actually, the thing to remember about genetics is you can never sequence someone in isolation. If you're sequencing a person, you're sequencing 50% of their parents and about 25% of their siblings. So the information that's uncovered has effects for the whole family. So people need to think quite carefully about, why they want to get tested and what they will do, especially if they find out something that's got implications. And that's not just health implications. You might go get sequenced and find out the person you thought was your father isn't your father, because that's actually, this type of testing is sometimes throwing up those types of things that people didn't realize, or that they didn't know they were adopted. So it's kind of like, it can shed light on a lot of things that people weren't aware of. And it's kind of like making sure you want to get all the answers you might get. And again, different people will have different opinions, but yes, I think quite often with babies, unless you think they're going to have something that's going to affect them in childhood that can be prevented with certain medicines or diets, or, you can check babies for certain conditions that mean if they don't eat something, they'll be much healthier than if they do, or to supplement diets. But if it's not going to affect them in the first 18 years, I think we need to be quite careful and let people make decisions about their own genetic information.
Fran's just found me a great link that Genomics England is about to start trialing this. So it's sort of not frontline medicine at the moment. It's sort of being trialed. And again, I'll pop another couple links from Genomics England on what the public thinks. So again, this is an area that we're not quite there yet. So about now is the time that as the public, we should be thinking about whether it should be a thing, because, especially if in the UK, if it's NHS money, then, is that a good use? Do people want it? All of those sorts of questions.
Exactly. It's the type of thing where we need more people involved, because it's like, I think in the States, the police have accessed some of these, these companies that you can get yourself sequenced if you don't tick the right privacy boxes. In cases like the US, they use that to find serial killers, or and it's a question of, do you want your information to be used that way? And just be aware of how it can be used if you get yourself sequenced. But on the other hand, in the UK, there's been a court case about somebody who found, a person found out they had one of these genetic diseases. So there was a 50% chance of their children having it. And they decided to not tell a child who was pregnant. So did that daughter have the right to know and to make a decision about the pregnancy? Or did the parent, because it's their health information, have the right to keep it from the daughter? So, these are the types of questions where we need to make a decision, especially for doctors. We used to always say doctor-patient confidentiality. Does that change when genetics are involved and the information revealed has health implications for family members? And these are the types of things we need to make decisions on, because now we have this technology. How do people want it to be used or not used?
We've got a really good activity around this, actually. I'll pop in the chat box now, called Genome Generation. So, no matter your age, it was sort of developed for upper high school students, but whatever age you are, it goes through a lot of these very sort of classic cases of, should your family know? Should you be allowed to find out? All of those sorts of questions, and shows you a lot of the sort of pros and cons. So if this is something you're interested in, it's a great resource that just gets you thinking about perspectives that maybe you haven't thought of, because if you've not got a genetic condition, you might not have thought of the pros and cons of finding out. So it's quite a useful one for sort of thinking through and talking through those sorts of thoughts. We're coming up to the end of our session. I'm just scrolling through to see if we've missed any questions, and I think we have covered pretty much all of them. So thank you everyone for joining us. Barry, thank you so much for giving us your time this evening. We've gone through viruses, we've gone through genomic surveillance, we've gone all the way up to sort of human genome editing. So thank you for answering so many questions related to your sort of work and career. And there's been a couple of lovely thank yous in the chat box as well. So thank you very much for joining us. I will just share my screen again if I can find the right button.
Well, thank you for the good questions.
Yeah, they had some absolutely awesome questions this evening. So hopefully you can all see this. So the next session we'll be hosting on this will be in about a month's time. We'll be looking at the topic of CRISPR. So we'll be looking at how CRISPR-Cas9 works, which is a genome editing sort of technology. And I'm sure we'll get onto similar questions of just because we can, should we? So if you're interested in joining that, there's a link for that on the screen at the moment, but you'll also get an email tomorrow from Eventbrite, well, through Eventbrite from us, with the recording for this video. So if you missed any of it, don't worry. The link for the next one, and we've also got like a learning pack that goes with this session. So if you're interested in any of the stuff that we covered, there'll be loads of more links that you can use to sort of delve into the topic more as well. So thank you so much everyone for joining. Obviously, Valerie, thank you so much for your time, and hopefully we will see you again in about a month.